Okay, next up we have Marc Hummersone from Astrea Bioseparations.
So first of all, thank you for the opportunity to speak to you all today. It's great to see so many faces that I recognise in the crowd. And what I'm here to tell you about is how we at Astrea are reimagining the future of cell and gene therapy purification. And this is a big part of everything that we're doing today because we've launched a new product, which I'm going to tell you about today. And it builds on the platform that we are working on at Astrea Bioseparations. So first of all, who are we? Let me walk you through the agenda, and I can tell you I'm going to introduce you to Astrea Bioseparations. I'm going to tell you about the fit-for-purpose solutions that we're developing in this space. And I'm going to walk you through a case study around lentiviral vector purification, and then give you a vision of where we're going to go in the future. So, who are we? At Astrea, we have a long history in bioseparations. We have a history that spans more than 30 years. We deliver products and services to the customer in the bioseparation space. We have a fantastic ligand discovery platform that are around synthetic ligands, and more recently we've added to that with protein-based Affimer ligand technology. We have manufacturing capability where we're able to manufacture single batches up to 1,000 litres. We have an overall capacity of around about 35,000 litres production capacity of resins per year. And we can do that in a clean room facility, which means that we're able to support customers around their regulatory support needs so that they can position their products in an FDA-approved process. And with that in mind, we have 21 products that are in an FDA-approved process. So we know what we're doing, essentially. We have a lot of expertise in resins, but what I'm going to tell you about is fiber. Before I do, however, I want you to know that we have a global presence. In Cambridge, in the UK, we have our research hub, but we also have manufacturing capability on the Isle of Man in the UK as well. We have manufacturing capability in Canada, in Joliet, and we also have Astrea presence in the US, in Boston, not too far from here, and we're building on that presence too. And throughout APAC, and we've got India, Poland, China, Israel, Ireland, Indonesia, and Japan, as well as other areas in APAC, we have a global presence. So what is Astrea Adept? It's a nanofiber technology, and this is going to be a game changer, particularly in the area of cell and gene therapy. But let me compare and contrast with resin-based technology. Legacy sorbents that you can see here in front of you, based on bead technology, have a porous network. And that means that for the larger modalities, those large modalities can't permeate into the pores. That results in reduced yield, long processing time, and genuine inefficiency. We've repurposed products that were made for other reasons, other markets. We've repurposed them for cell and gene. But the Astrea Adept fiber-based solution is one that has a huge surface area and allows us to put a high concentration of ligands per unit volume of sorbent. It allows a tremendously fast flow rate. And as such, we can cycle very quickly, and that gives us a reduced footprint. We don't have to rely on a passive diffusion event that you do require for resins. And as such, we typically have residence time of down to sub 1 second, which means that we can go fast. To give you an example of the chromatographic performance of these fiber-based materials, you can see in front of you, we have a chromatogram where we are binding BSA on a weak anion exchange media. And we can typically get binding capacities of BSA of around about 120 megamils, which is fantastic in its own right. But on the right-hand side, you can see we're running that chromatography at 60 membrane volumes a minute. And we're able to do this with a very low back pressure as well. So that means that we can have a residence time of sub 1 second. So we can do things fast, we can do them efficiently, and we are able to compress your processes so that your productivity increases with the technology. Now, the material itself is a true composite. We've electrospun more than one polymer. One of the polymers allows us to add chemistry to the surface of the nanofibers, and the other one is there for strength. On the right-hand side, you can see when we look at those polymers in isolation, the purple line is where we're trying to stretch just cellulosic materials. And you put a lot of tension into those materials, and they don't stretch very much, and then they fail. The black line is our other special polymer that gives us an elasticity of the materials. And then when we combine them, we can have a strength that's double that of cellulose on its own, but an increased flexibility, which means that we have a tremendous design space in which we can put these materials for our larger devices and beyond. So I've told you about the material, and that we describe that as an electrospun nanofiber, but it's going into a product that we call LentiHero. And it's all about the purification of lentiviral samples. It's the high throughput purification of lentiviral particles, where you've got a high number of samples, and this is on a discovery scale. So typically, what we would find is that we have many, many samples. They're normally quite small, and when we try and purify them and mess with them to get them into a pure form, we're damaging them. We're reducing the sample size. We're compromising the quality of those samples. And so typically on a research scale, you'll end up with very small samples of very low quality. And as such, when you're testing those samples, you can often find false negatives because the virus isn't behaving the way that you want it to and it's damaged. So the key challenges that we have addressed with this new product are around overcoming the low viral particle recovery and the low processing throughput. And there's a pretty poor contamination profile on the research scale. And we have addressed all of those things in the LentiHero product, and which I mentioned we've launched that today. So the LentiHero product it's a fantastic device. You don't even need enabling technology in terms of ectosystems or any other pumping. You just need a centrifuge to make this stuff work. And it's in a spin column format. So that in 5 easy steps, you can prepare your column, apply your sample, wash and elute, and it'll give you a highly pure sample on a research scale. And if you need to scale out on a research scale, you can do that by applying these to a vacuum manifold as well. So you have flexibility for the user, and you don't have to be a chromatography expert to make this stuff work. So we have superior bioprocessing capabilities, and what we're able to do as well, because the material is so different from regular resins that you all know of, is that we can do our purification with and without serum. And you can see the graph on the right-hand side here, with and without serum gives us good recovery of active viral particles in a pure form. It's cutting down the number of manipulation steps that you have to do with your R&D sample in order to purify it to give you effective viral particles. And here's some more data that I can show you how well the particles survive the purification process. On the left-hand side, we've got lentivirus specific detection of the GAG protein, which is something that's heavily involved in all of the transduction steps. And we find that it's there and it's competent after the purification. The TEM images as well show you that our viral particles are not damaged. They are in great condition because they've only been residents in their chromatography bed for a fraction of a second. And actually, when we've purified, we can see from the transduction data that they're doing what they should do. They're transferring their genetic material. These viral particles are pure, they're active, and they're doing exactly what we want them to do. And you know what? We can get very good recoveries using this technology above and beyond what is normally achievable with regular resin-based purification technologies. So the purity is, we have added benefits of using nanofiber technology. And the nanofiber material that's in the spin column itself has the added bonus of not just giving us pure viral particles, we actually see a significant reduction in the HCP load of our samples as well. And typically, the reduction in the HCP load is around about 95%. And as such, it's further reducing the amount of manipulations that you need to do with your lentiviral prep. And the SDS page data here can show you that after the elution, there is virtually no HCP present. It's a significant reduction. We've quantified it, and it's around about 95% reduction. So it's an added benefit of the purification step. You don't have to mess around with it so much so that your viral particles remain competent. This means, in turn, that you've got less false negatives because your viral particles are good, they're competent, they're healthy. This means that when you're making decisions at a research scale for each of the preparations that the Lenti-Hero allows you to do, you will get a true understanding of what your hit rate is for your discovery process. So this is really, really focusing down your abilities to choose the correct molecule. So in a nutshell, what are we offering with the Lenti-Hero product? And first of all, we're able to reduce the overall process time significantly. It takes no longer than a spin on a centrifuge to make this stuff happen, to give you your pure samples. We don't need very much enabling technology. You only need a centrifuge, as I say. On a research scale, we can scale out. We can take multiples of these tubes and put them on a vacuum manifold, and that gives you the ability to purify more viral particles effectively on a research scale. And as such, we're able to compress your processes. We can reduce time. We're increasing the recovery that is available for your viral particles. And they're able to really supercharge your lentiviral discovery activities. And the fantastic thing is, because it's in a centrifuge, because you're using a centrifuge only and using a spin column format, you don't have to be a chromatography expert. So the secret is in the name. We're actually making the user the hero. You take your sample, you put it in your tube, you follow the 5 steps, and you get pure lentivirus sample at the end, making you the hero in the process. So our first offering in this area is around small research device, but we're already working on delivering the future around nanofiber materials and how we're going to position it within the cell and gene therapy space. We're working with these high recovery rates in devices that will allow us to go from research to development and production and beyond. We have the capability to deliver on a manufacturing scale of Australia around resins, and we're looking to duplicate that know-how for fiber. This truly does represent the next generation of purification solutions for cell and gene therapy, and we're at the forefront of that technology. And we're working on larger devices, but actually the footprint is smaller. We can cycle so fast that we can carry out a large purification in a relatively short amount of time with small devices that run incredibly quickly. So I'd like to say, first of all, thank you very much for listening. Like my chromatography, we've made this fast. I'm sure you'll have plenty of questions. I'm more than happy to take questions from any of you. We have had our launch today at booth 901. Please come by and see us. If you see anybody with an Australia Adept t-shirt on, they'll be able to tell you more. And you can come and get an Australia Adept t-shirt and be a hero too. So thank you for your time. I appreciate it. And come and see us at booth 901. Thank you.
We do have some time for questions if anyone has any.
Hi, this is really, really interesting. I've spent, 16 hours where you purify out just a couple liters of AAV soup, so this is really cool. Yep. What's the total amount of time from when you start until you get your final product in your tube?
My apologies. It's very short. It's within minutes per spin on a centrifuge. So you have 5 basic steps. You condition your bed, you load your sample, you elute, you wash and then elute. How long does that take you to do on a centrifuge? It's not 16 hours, I can tell you that. Significantly shorter. So that really does exemplify process compression. You can do way more per unit volume of your time using this technology. So it's minutes, not hours. Great stuff.
So I'm inherently lazy, and I use fluid handling robot for as much as I can get away with. So I use, sorry, used to. So I prefer to use fluid handling robot. Is there any way that, or do you have any intention of making this automation friendly?
So if I just
Any intention of making it automation friendly?
Yeah, I was just about to repeat the question. Yes, we do. So the fantastic thing about the technology is that we can achieve an incredible amount of purification power with a small amount of our fiber material, which means that it's completely amenable for a plate format. That's something that we are very, very interested in pursuing, which would lend itself to a liquid handling platform such as the ones that you've been describing. And that has benefits to the user because you can optimize your conditions really, really quickly. So yes, absolutely 100%. Okay,
If we don't have any more questions, we're good. Thanks, Mark.
great stuff. Thank you very much.